DNA replication is the process of producing two identical replicas from one original DNA molecule, enabling cells to replicate themselves in a quick and efficient manner. And the structure of DNA chain helps in speed and accuracy of the replication. DNA is a double-stranded molecule made by two strands of nucleotides bonded by hydrogen bonds. Nucleotides are molecules that consist of deoxyribose sugar, a phosphate, and one of four nitrogenous bases: cytosine, adenine, thymine, and guanine. Cytosine forms triple hydrogen bond with guanine, while adenine forms double hydrogen bond with thymine, with both of these bonds being referred to as a complementary base pairing. The phosphate and deoxyribose sugars alternate to form the strand. The double-stranded DNA has one strand headed in 5-3 direction, while other in 3-5 direction, forming the anti-parallel nature of the DNA. The DNA replication begins at a point called the origin of replication, which then proceeds to denote the area of replication known as the replication fork. Once the fork is established, numerous enzymes are called to work, as they are essential for the replication to follow through. Helicase separates the double strand into two strands, which provide a template for creating a new strand of DNA. They are enforced and stabilized by single-stranded binding proteins until new nucleotides are connected by DNA polymerase to the separated strands. However, before DNA polymerase does so, enzyme primase has to connect short pieces of RNA called primer. This is a starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and makes the new strand of DNA. The replication of both strands happens at the same time, one of which is replicated in continuous and other in discontinuous manner. That means that the strand, referred to as the leading strand, oriented in the 3-5 direction, is being connected with nucleotides continuously towards the expanding fork. The other strand, called lagging strand, which is then connected at discontinuous manner because DNA polymerase can only add DNA bases from 5-3 direction. DNA polymerase then fills in the gap between the primers in 5-3 direction by short rows of DNA bases, the Okazaki fragments. This process is repeated again and again. After finishing this process, the enzyme exonuclease removes all the RNA primers from both strands of DNA. The gaps are filled by DNA polymerase with DNA bases. In the end, enzyme DNA ligase seals up the fragments of DNA in both strands to form a nice smooth double strand. This entire process ends when the entire DNA sequence has been replicated and therefore separated into identical DNA sequences, enabling the cells to divide and create identical replicas for themselves. The replication is described as semi-conservative because each DNA is made from one old, conserved strand and one new strand.